All right, well, let's get started for the second session this morning so we can get to lunch more or less on time. We'll see how the time goes, and I have a software demo that I was going to do for part of it, but we can do that over lunch like we did yesterday if we run a little bit behind on time. So I don't know how many of you, I didn't introduce myself to you yesterday, so I'll just say I'm Jim Fader from the University of Pittsburgh Department of Computational and Systems Biology, part of the BIOS project with Marcus and Tom and Marcus, and then we have a bunch of other people from that project here. I won't list all the names because I'll forget someone undoubtedly, and I have some slides about that coming up. So I'm going to talk about rule-based modeling and how we model complex biochemical systems and networks using tools that I've been developing in my lab for a while, BioNetGen and RuleBender, which is a graphical interface for BioNetGen, and which we're actively working on linking to MCell and providing basically more robust techniques for handling molecular complexity, which I'll talk about in a little bit. Marcus gave an introduction to the BTRC yesterday, this high-performance computing for multi-scale modeling of biological systems. It's a center that links the Pittsburgh Supercomputing Center, University of Pittsburgh, Carnegie Mellon here locally, and then Salk Institute in San Diego. Oh, it has. All right. And did you already present this slide, Marcus? I did. Okay. Okay. So I just wanted to emphasize the multi-scale aspect of this endeavor and the sort of the overarching ambitions that we have to study in detail the spatial organization and temporal evolution of complex biological signaling systems. So there's a big focus on neuroscience, and many of you are coming from that community, but there's also within the project interest in signal transduction over a wide variety of biological systems, from bacteria up through so cancer biology, for example, which we'll see some examples from in this talk. Yes, this is Pittsburgh, you all know. This sign is just down the street in case you haven't seen it yet. That's the BST-3, where the part of the medical school at the University of Pittsburgh, where the Computational and Systems Biology department is. This is the Gates-Hillman Center, which houses the School of Computer Science at Carnegie Mellon. It's a fairly new, beautiful building. If you'd like to go see, if you're interested in architecture, modern design of buildings, I would invite you to go check that out. It's kind of interesting, funky stuff, including like a helical staircase. And then this is Salk Institute, of course, on a rare cloudy day in San Diego. And then this is somehow all of us. I don't even remember posing for this picture. Okay, and the idea here, there are three main components of the center that are organized around different, basically computing or simulation technologies and biological systems. At the lowest scale of individual molecules, proteins, this is a complex assembly in a membrane, I think. I don't know what that is. This I know what it is. This is a reconstruction of a hippocampal neuropil. There's a huge number of different synapses represented in this very dense region of the brain. And this is actually the reconstruction of this geometry that was done by Tom and company. And this is a simulatable MCell model of this system. And then up to the level of neural circuitry, there are members of the center who are working actively to reconstruct large-scale networks of neuronal connections from electron microscopy data, huge data sets that are modeled or that are reconstructed on some of the architecture that's depicted on various posters that you'll see around PSC.
Sorry, those are micrometers too.
These are the scales are you asking about?
It says millimeter.
This is millimeter scale, yeah. This is unbelievable.
No, micrometer.
Oh, these are micrometers. Oh, okay. Yeah, I didn't make this slide. I take no responsibility for that, but I should have caught it. You're right. They are millimeters, actually. 0.4, 0.4, and 0.05. Okay. So, sorry? We should correct that, because this is the slide deck that came from the boss. So I didn't make it. But you can see we're going from roughly from nanometer scale, the scale of individual molecules, individual proteins, and almost to the level of individual water molecules, all the way up to large networks of intracellular connections. M-cell, of course, lives in this space, and we're trying to connect this molecular scale all the way up to this cellular scale. And one of the goals, M-cell is one of the sort of driving technologies of the center. One of the goals is to increase our ability to deal with this level of complexity and get information from structural modeling, for example. So all atom MD simulations can be used to inform the biochemical models that we put into M-cell, and ultimately to develop models of cellular systems where we have many different cell types interacting with each other through complex biochemical circuitry. So we're trying to integrate information across scales. And the organization of the project looks something like this. It's our first example of a complex network today that we're going to be talking about, but only very briefly. So there are three main what are called technology, research, and development components of this center that are organized along these scale boundaries. At the highest level, we have the component that's involved in imaging and reconstruction of spatial distributions from within cells to within tissues. M-cell, the cellular modeling component, is here at this level, at this TRND2 level, and then at the molecular level, we have TRND1, which is a collection of different groups that are doing molecular to supramolecular modeling. And then driving the development of all this technology are various collaborations and so-called driving biomedical projects, these DBPs, that involve targeted experimental efforts for various problems, trafficking of different receptors in neurons, signaling at in the postsynaptic density of neurons. The DBP4 is T-cell signaling, actually. Okay. So we also talked briefly about, so now we're focusing in on the TRND2 level, the level of the spatial, the cellular modeling. Marcus presented this a little bit yesterday. There are a number of different tools for spatial modeling of biochemical cellular systems, M-cell being one of them, and we tried to map out the sort of different levels of resolution of these different techniques, from virtual cell, which handles primarily partial differential equations, down to the particle-based simulators, particularly Smolden and M-cell. And what we're trying to do in our development of M-cell as part of this project is really to increase the ability to handle not only the spatial complexity within M-cell, but also the different levels of molecular complexity that can be represented. And we'll see some examples of when this becomes a really challenging problem, actually in the next slide. Okay, so if we look at what's going on, for example, the kind of signal transduction that occurs in postsynaptic density, where you have various neurotransmitters triggering different receptors that are in the cell membrane here. These trigger a very complex biochemical cascade of reactions involving the assembly of large macromolecular complexes or molecular machines in this region. And when we want to model these in terms of a biochemical reaction network, there's this huge molecular complexity that we have to deal with. So, for example, the CAMK2 kinase, which is one of the central molecular signaling hubs that's present here, it's present as a dodecamer. Okay, so there are 12 subunits of that complex. These subunits, each one of them has kinase activity, each one of them has phosphorylation sites. And so the state space, the possible number of different configurations of just this one 12-member cluster is astronomical, and it's represented here. So it goes exponentially. Each subunit here has a possible, in the current model that's being developed by Tom and collaborators at Caltech, there are 40 different states of each monomer, And because there are 12 monomers, there's something like 40 to the 12 possible different states of that dodecameric complex. And if you had to, you may have had some experience yesterday of writing down a reaction network in MCell. If you had to write down equations or reactions governing each of these individual states, you would be pretty much out of steam.
Reactions is 10 to the 21st.
Yeah, it's something much larger than this even. You simply can't do it. So we know that there are limits. And obviously, when one is developing these kinds of reaction network models, the first approach you're going to take is to abstract this. Okay, let's just consider that this is a receptor, and then this receptor activates this complex, and so you have a receptor state, and it can be inactive or active, and then you have the complex, and it's inactive or active, and so on and so forth. But this is a little bit unsatisfying because it really doesn't represent what we know about the biochemical complexity, and eventually you're going to hit up against these kinds of barriers, and it comes up sooner than you think. So let's talk a little bit about, okay, so what's the standard way that you write a reaction network model for one of these systems? So I have nice pictures from our studies of receptors in the immune system. This is the IgE receptor. IgE is an antibody that's produced largely as a component of allergic responses. It binds selectively to various foreign, usually often nominally harmless foreign substances that are, we breathe in, maybe from pollen or from dust mites or you name it, some food that you eat, such as an apple or a berry, and some proteins that are components of those different environmental substances are recognized with high specificity by these the binding sites on this antibody. What I'm showing here, this IgE dimer, is actually something that doesn't exist in your body. That's something that only exists in the laboratory experiments that we're doing to characterize the system. But the IgE is floating in your bloodstream. It recognizes different substances. It binds to them. And you form basically aggregates of the receptors, this high affinity receptor for IgE called Fc epsilon R1. When you aggregate this receptor, the little bumps get phosphorylated. Those little phosphorylated bumps recruit other kinase molecules that are present in the cells that contain these receptors, primarily mast cells and basophils. And they trigger a cascade of biochemical reactions that lead you to do things like produce histamine and other kinds of inflammatory mediators that make your life miserable during this time of year. So this signaling system, anyway, it's just a representative of any possible biochemical cascade where you have a ligand, receptors, kinases, and various modifications of all of their states. What we know about the biochemistry of the system is typically represented in terms of various kinds of interactions. Binding interaction between the ligand and the receptor. The ligand in this case has two binding sites for the receptor, so it can make dimers of the receptor. The dimeric form of the receptor is the active one. In the dimeric form, you can have, I'm going to give up, you can have phosphorylation reactions. So when you have a dimer and you have a kinase that's present in the dimer, it can transphosphorylate the receptor. These phosphorylation sites then can recruit an additional kinase molecule. They also serve as a higher affinity binding site for this yellow kinase, which forms a kind of a positive feedback in this system. So it's very easy to represent the biochemistry in terms of these simple interactions, simple arrow diagrams. The kinase associates with the receptor in both unphosphorylated and phosphorylated state. There's a dephosphorylation reaction that affects all of the different sites in our molecule, in our model, and so on and so forth. So this is, seems like a fairly simple system. And we'll see that it's actually not. So the next step would be to then, in a standard approach, and if we're just doing the standard thing that we would do in MCell, we need to figure out, okay, given these components and these interactions, what is the reaction network that's implied by this? And if we're using MATLAB to do our modeling, as many people in this field do, we would need to construct a set of differential equations where x here represent the concentrations of the species, of all the different possible species in the system. This S is a stoichiometry matrix, is a way of basically describing the biochemical reactions, translating the biochemical reactions into a set of differential equations, and these V's are the fluxes of the individual reactions. In M-cell, we write a series of reactions, and it takes care of, it doesn't actually construct differential equations, but it takes care of effectively making doing the computations to compute what reactions occur. Okay, but and a typical reaction network could be encoded in, for example, systems biology markup language, SBML, which many of you may have had contact with. Okay, and then the next step, once we have our equations or a reaction network, we need to get parameters for the different components of the system. So here we had 4 different molecular components. We would need to know their concentrations. One way to represent those would be in terms of copy numbers per cell, the concentration of the ligand. We need some way to relate these to use a consistent set of units for our equations to get all of these together. We'll see that's a non-trivial problem later on. Okay, and then assuming we can do all of this, we could write our MATLAB file. We could construct an SBML file using any one of the number of tools to make reaction networks, and then we can do simulations. We could use, if we're assuming everything is well mixed, we can use our ordinary differential equations. We can use stochastic simulations that correspond basically to these differential equations in the discrete limit, but where we still assume that diffusion is fast, that the system is mixing itself. We're not going to consider spatial effects. And then these are spatial modeling methods, partial differential equations, Brownian dynamics, and M-cell is, of course, down here. But the reaction network, the fundamental equations that we need to describe this biochemistry is all the same. The language of this can get fed to differential equations, stochastic simulations, or spatial simulations, spatial differential equations or spatial stochastic. Okay? It's all the same basic underlying network. Different assumptions are going to be made about how we actually simulate the molecules in space and time here, or simulate the concentrations in space and time here. And if all of this seems a little bit abstract, we'll make it quite concrete, so don't worry too much about that. So here's what I mean when I say translate our reaction model, our physical interactions, into a set of reactions and differential equations. So if you've seen this before, just bear with me for. We're just going to do a very quick flyover for this. But the idea is, okay, so we have ligand binds to receptor. We can represent that as a simple reaction. R plus L goes to RL. And if we assume that we have elementary mass action kinetics, we can write a rate constant K1, and the reaction velocity, the rate, the number of reactions, or the concentration of reactions per second or per unit time is just given by this rate constant K1. Not sure what this is, some artifact here, times the concentration of R times the concentration of L. So does that make sense to everyone here? Okay. Simple enough. If we have this mass action rate law, then we can write differential equations for the rate of change of all of the different species in our system. Here we have 3 so far. We have the free ligand, the free receptor free ligand, and the bound ligand receptor complex, which I call B here. But here I call it RL. And what we see is that the rate of change, the term that comes from this particular reaction, is just, for the case of receptors being consumed by this reaction, so there's a minus V1. This minus 1 is the stoichiometry of this species 1, species R, in reaction 1. So if you remember from the previous slide, I had this matrix S, this quantity S. That stoichiometry matrix comes from the coefficients that go in front of the reaction fluxes here. Okay, and so R is consumed, L is consumed, we have a minus V1 here, and RL is produced by this reaction, so we have a plus 1 V1. If two RL molecules were produced, we'd have a 2 here, for example. Or if let's just say we needed to bind 2 ligand molecules to make this bound complex, we would have a minus 2. that might also affect our mass action kinetics as well. But we're not going to worry about that for right now. So this is what I mean by writing down our reaction network. Our reaction network consists of a set of reactions that look like this, and we know that there's an automatic way, assuming we know what the form of the rate laws is. They can be arbitrarily complex mathematical functions of the species concentrations, but typically we'll assume for most reactions they obey some form of mass action. We can write down a set of differential equations. If we know the initial concentrations, we can solve the differential equations, and that's a simulation model. We can solve differential equations over time, and that gives us the concentrations of everything over time, and we can change the parameters, and we can investigate the behavior of that system. Okay, so this scales to a certain degree. What I've shown here is the Holodenko model of epidermal growth factor receptor signaling. So here's the ligand here, here's the receptor molecule. Ligand binds receptor, it forms this ligand receptor complex. That ligand receptor complex dimerizes, it gets phosphorylated. The phosphorylated receptor can bind different molecules, Schick, Grab2, PLC gamma, and activate more stuff. Okay, and typical process is write down some kind of a. They actually, before they wrote down this diagram, probably mapped out what molecules the receptor could bind to, Grab2 and Schick, and PLC gamma, and then they mapped out the different states they wanted to consider, and eventually they constructed this network diagram. And from that, you can write down a set of biochemical reactions. From those reactions, you can derive your rate equation. So that's what they've done here. So for each reaction in the system, they actually group the forward and reverse reactions together. So here we have receptor binding to the ligand. The forward rate is K1 times receptor concentration times ligand concentration, and then that bound complex breaks up with rate K minus 1 times the concentration of the ligand receptor complex. Here are the differential equations they derive for the system. There are 22, and there are 25 different reactions for this system, okay, that are encoded by this diagram. So we started with an interaction model, and then we went through this process of constructing a network diagram, constructing all the equations, putting them into some form of modeling software. So what's the problem with this? This is great. This is what many of you will do when constructing a model of a particular receptor system that's going to couple to some response in the cells that you're interested in. The problem comes that typically we know a lot about the interactions of signaling molecules that are in systems. This is a diagram of the epidermal growth factor receptor system. We have the EGF ligand, the EGFR molecule. It turns out it has a number of different phosphorylation sites, and to those sites it is known that the Grab2 adapter and Schick can bind to a number of different ones. It's not very clear from the literature how many sites you can have bound in one receptor molecule, for example. How many Grab2 and Schick you can have potentially. There are 4 different sites, so you could have up to 4 different sites bound. And there are going to be a huge number of different combinations of Schick and Grab2 that can bind to 4 sites if you let all 4 sites be bound at once. But we don't really know exactly what the rules are that say how many you can bind. But if we take this diagram at face value, then it turns out from these 5 different proteins and the 20 interactions that we can write down, there are something like 170,000 species. So there are 170,000 differential equations that you'd have to write down to map out this diagram, which is not something you want to do by hand. So if your research advisor said, I want a model of this system, I don't want you to make any approximations or any assumptions about how many Grab2 and Schick can bind, assuming that you didn't just ignore that and go on and do whatever you're going to do anyway, you'd write a program to do that. So some time ago, when we were developing BioNetGen and we were trying to study the IgE receptor, that's exactly what we did. We started writing programs to generate networks based on diagrams like this. So instead of writing down these equations by hand, we started to have a computer generate those equations. And it turns out that biochemical signaling is complicated, but it's really the composition of a lot of interactions of basically the same type. Only a few different things really have to be considered to generate the network complexity. So it's very easy to teach a computer to design an algorithm that will generate this network for you. To get all the symmetry and properties right for such a generator is not trivial, but it's much better than writing it by hand. So if we were, this system that we started with, this IgE receptor system, so these components, these interactions, there are 354 equations. There are 354 different chemical species that arise from this system, and about 3,600 reactions. And that was small enough that back in the beginning we thought we could do that all by, well, I never thought I could do that by hand. But the people that I was working for thought they could do it by hand, and they had a committee of 6 people to write the equations. Really. And the youngest member of the team, this was before I got there, realized that these guys who were doing this, they were never going to get all the factors of 2 right in those equations. Never, ever. It was never going to converge. And so he wrote a program to do that himself and got most of the factors of 2, I think all of them right eventually. And much faster than a group of 6 people could agree on the coefficients for 3,680 reactions. And so that was the beginning of our effort to do this. Okay, so we want to write these, we want to have a computer program do this for us. Another example, if we expand out, if we look at different, this is very typical of many different kinds of receptors, both in growth factor signaling, in cytokine or immune receptor signaling, in neuronal receptor signaling. You get this huge combinatorial complexity that comes from all these different phosphorylation sites binding all this set of different molecules. And so just some bookkeeping is shown here. If for each of these sites we assume that there's 1 or 2 binding partners, you get really astronomical numbers. This is exponential, and you get really astronomical numbers of possible species. For the PDGF receptor, for example, there's 7 phosphorylation sites, so there's 2 to the 7th different phosphorylation states of the receptor. But because you can bind at least 1 protein at each of those sites, you have at least 3 or 4 states at each of them, and you get 4,000 different monomer states. All of these receptors are active as dimers, at least at a minimum. Higher order oligomers are possible. So you just have huge numbers of species. And so it's really typical. This is not just the pathology of CAMK2 or the pathology of EGFR or the pathology of the T cell receptor or of the B cell receptor, or it's everywhere. Okay, it's true in bacterial signaling as well, although that complexity is usually abstracted away conveniently. Okay, all right. So the key idea of rule-based modeling is that we want to make a translation of this diagram where we translate what interactions are present here, but we don't try to generate the network. We let the computer do that part for us. How do we do that? So we want to break down these complex molecules into their essential building blocks. What I've shown here is the basic building blocks of a rule. So a rule is going to describe the interactions of any number of molecules and their components. And it basically says when a reaction can happen. So the simplest kind of a rule that we can make between 2 different molecules is where one molecule binds to another. A binds to B. So here's our first rule. If we have a simple edge between 2 proteins in a network diagram, we assume that there's got to be some part of those proteins that is responsible for the binding interface, right? And we give names to those binding, the components of the binding interface. If we don't know anything about the actual components, they're not, for example, conserved, highly conserved domains that have a name like an SH2 or some other. We might just call them B and A where little b represents the binding site on the A molecule for the big B molecule, and vice versa. So we're just basically naming these, we call that cognate binding site names. And our rule says that B binds to A. The B site of A binds to the A site of B. And this is it. This could be a simple biochemical rule for a binding interaction. It has no context. It says only that this site has to be free, and this site has to be free, and then the two sites can bind to each other. And the basic formalism, we always want to have at most one bond per individual site, because we're representing, again, here some kind of a physical interface. So if that interface is occupied through binding, then we can't bind something else there as well. So this simple rule for binding between A and B actually has many possible consequences in a complex network. There are many different forms of the A molecule, for example, and many different forms of the B molecule, which they can have other components. So this A molecule had a binding site for B here, but it may have another binding site for ligand, a phosphorylation site. Maybe that phosphorylation site binds something else. Probably has 15 phosphorylation sites, only 8 of which are recognized as being important. Okay, so we don't know how much complexity there is under the surface, but we want this rule to say that A and B, unless we know otherwise, these guys can bind to each other as long as there's a free binding site. And so all of these different possible molecular species in our system are candidates for binding to a B molecule, and all of these B molecules are candidates for binding to an A molecule because they have the property that their binding site is free, all of these guys here. Okay? So this rule then represents many possible binding interactions. If I wanted to generate all of the different possible reactions in the network comprised of this set of A's and B's, I would have to take all the combinations. And again, that's something you'd much rather have the computer do than do yourself. That's the basic idea. So far we have our binding rule, and the idea here is that rules have two different parts. One part is what action is taking place. So here we're going to form a bond. But now we can be more specific. We can say this bond can form under a certain set of conditions. Maybe this binding site on A is only exposed when a ligand binds to its ligand binding site, and there's a conformational change that occurs that exposes this binding site. So we could have a rule that requires, for example, the ligand binding site of A to be bound before this interaction can take place. That additional information is called the context for the rule. That allows us to make rules that are more specific. Here I've added another piece of context. I've said that this other phosphorylation site on A has to be in the unphosphorylated form. Okay, and right. So the idea of the context is these are additional requirements that the reactants must satisfy in order for the transformation that's carried out by the rule to take place. So in order for A to bind B, A has to be unphosphorylated and bound to ligand. That's what this simple rule says. And the idea is that we can then refine rules as we get more biochemical information and encode that information directly. So, right. So for example, if there's cooperativity or there's allostery, this would be an example, possibly an example of an allosteric effect where the ligand binding changes the state of this binding component so that the receptor can bind, so that the B molecule can bind. Alrighty. Okay, so what do I want to say here? What we can see from looking at reaction networks from this perspective is that rules are sort of generalized the standard picture. In other words, here, if I have a complex network with many different possible states, then if what I know is that A interacts with B and it forms a bond, if I were to somehow select only certain varieties of A to bind to certain varieties of B, or one particular instance of A to bind to one particular instance of B, I'm making additional assumptions, right? I'm saying that for some reason all of these states are unsuitable, but this one and this one are suitable. So one way of thinking about sort of standard reaction networks is that they represent basically a highly specific set of assumptions made on a more general class of interactions. And these assumptions are really hidden from view. And we'll actually see in the lab today an example. We take a model from the literature. We basically extract the binding sites and phosphorylation sites of the components of that model. And then we look at if you look at the rule set that's generated by doing that, you'll see things like this, where only one particular state of the reactant species is selected, but there could be many other possible ones in that system that aren't considered in the network. So typically, the modeler is making assumptions like, the phosphorylated form of A that's not bound to ligand doesn't exist in my system. Because if ligand comes off, then A is not going to be phosphorylated. And sometimes that's a perfectly valid assumption, and other times it's not. One problem with that is that oftentimes this assumption was true under the original conditions for which that model was specified. You said, okay, I know that in the active form of this complex, the ligand's always bound. And so you made a model, and we eliminated this state. This phosphorylated state doesn't exist. But now we want to go back and model a situation, an experimental situation, where the property of the ligand was changed. Now, it may no longer be true that this state doesn't form. And if you use the original model that you developed, making that assumption, you may get wrong inferences from using that model compared to what you would see in your experiment. And the problem is not that you explicitly eliminated the state, it's that you forgot entirely that the model even made that assumption. Or you picked somebody else, you took this model out of the literature, and you didn't even realize that it made that assumption. Okay, and yeah, so we'll see more. We'll see some examples of that as we go along. So here's another example, just another example of a rule. I talked about binding already. Another kind of a rule is what I call here a state change. This is a phosphorylation reaction where the A is being phosphorylated here, presumably by some kind of autocatalytic activity, because I haven't specified any other molecule in the system. So there's no kinase enzyme here explicitly represented that's causing this action. We're just saying that it's happening because of whatever additional context I provided. In this case, when the ligand binds to the A molecule, something is activated in A that allows A to become phosphorylated. Okay. So that's another example of context. All right. So we can come back to this, this picture, and we can see that we could, in principle, write down rules for each of the interaction links that's shown in the diagram. And potentially, there might be more information. We might have more information about some of these interactions than is actually shown by just representing the individual links. The links basically represent the reaction centers. And additional context is mostly, for the most part, left out of this this diagram. But that's not so important here. Okay, so let's review what the basic protocol is then. We start now with a system of molecules and interactions, and we encode those instead of manually constructing a set of differential equations. We represent these as a set of structured molecules. I used a graphical representation before. Here I'm introducing the basic BioNetGen text-based description. So here we have a molecule and two components, two sites A that are both called A, and we can do that. We can make multiple copies of the same thing. So this represents an IgE dimer, for example, with two A sites. This is our receptor, which has an A, a B, and a G2 site. The G2 represents the gamma-2 homodimer, which is part of this multimeric receptor assembly. And what's shown here, and we'll see more of the syntax as we go along, what's shown here is the fact that the B site, this B component, has two different possible states, a U state and a P state, representing unphosphorylated and phosphorylated forms of that component. And we see another molecule here, SIC, has a tandem SH2 domain, another tyrosine that's an unphosphorylated and phosphorylated state has an unphosphorylated and phosphorylated state, and same for this A tyrosine. This is A stands for activation loop here. So we've used a condensed representation of these the components of the molecule. We could use more descriptive forms if we wanted to. The names can be arbitrarily complicated, but sometimes that can obscure the simplicity of the reactions you're trying to represent. And then once we have the basic objects, the basic molecules that are the building blocks of the system, we can write their interactions in terms of rules. I've shown you some examples of simple binding rules and a simple phosphorylation rule. Here's a more complicated phosphorylation rule. So here the receptor B subunit is getting phosphorylated. It's going from U to P. But it requires, in order for this to happen, it requires that this receptor is in complex with another receptor that's in turn bound to a kinase molecule. And that kinase has to be bound in a particular way to the receptor. So this is all the context of the rule, and we can see that this rule can represent quite a bit of biochemical information. In the case of the IgE receptor system, we knew from various experimental lines of evidence that receptor aggregates could become transphosphorylated by kinases that were associated with that aggregate, but that the kinases did not appear to, for example, phosphorylate the tyrosines that were on the receptor to which that kinase molecule was attached. You could question how definitive the experiments were that gave rise to that information, but once you accept that that's the correct inference, then we can represent that inference in a rule in a very concise way. Okay. And then the rest of the procedure is basically the same. We need to parameterize these models with things like initial concentrations and rate constants for these different reaction processes. The approximate number of parameters we need to characterize a rule-based model is the same as the number of parameters we would need for an essentially equivalent reaction network model of these components and interactions. But basically the way we instead of having one reaction per interaction representing a whole bunch of different actual interactions in our network, we just have one rule that's parameterized by a single rate constant. And so, yeah, I'm not sure. Does that make sense to you? You're okay with that? So the key point is that basically the number of parameters is roughly comparable. If I start with this system that I want to model, I can take the rule-based approach, or I can write down sort of a very coarse-grained set of differential equations, and I'm going to have about the same number of parameters because I'm starting from the same knowledge about the system. Okay, and then once we have this description, we can use the computer to generate the appropriate reaction network to simulate ODEs or stochastics or spatial. And in fact, we can take our BioNetGen model, for example, export it as an SBML file that we can pass to other tools or MATLAB, or we can export it as MDL. That's actually work that's in progress, and pass the reaction network that we generated to MCell and do simulations. Okay, so here's the basic model. Then we have a particular system. We frame that in terms of a set of molecules and interactions that we describe as rules, and then we take that model and we pass it to a simulator. And I'll talk about how we simulate rule-based models a little bit later on. Maybe actually what I'm going to do, I think, since I see the time, is I'll skip a little bit the details of how we specify models and come back to that. I want to just tell you a little bit about how we simulate these models. So kind of, yeah. So here's our rule-based model, molecules and reactions, and we can use this to represent many different kinds of systems. So just some examples are illustrated here, different classes of systems that we or collaborators or other people in the modeling community have developed detailed biochemical rule-based models for. We've tried to keep track of different publications of rule-based models in the literature, and we're working on compiling a comprehensive database of the models, which will be hopefully available pretty soon. So, okay, and you can check our website for some examples. But I wanted to talk a little bit, okay, and once we have such a model described, how do we go about simulating it? And here's the key idea. So what we did so far was we wrote our molecules and interactions, which I call here objects and rules. We have a program called BioNetGen. So this was in the BioNetGen language. We passed this model description to a simulator, an interpreter, basically. The simplest way, if we want to do a well-mixed simulation of a biochemical system, the standard procedure would be to take that set of molecules and interactions and generate the full reaction network that's implied by that specification. So find, in an automated way, all the possible species and all the ways that they can react with each other to make a reaction network. This reaction network can be described in a number of different ways. It can be exported, for example, as an SBML file. It could be exported to MATLAB, Mathematica, whatever. Then you can pass that to a simulator, that description to a simulator of some sort. That simulator could be basically an ODE solver, or it could be a stochastic simulation algorithm of some sort, such as the Gillespie algorithm. In either case, you're going to get the time course, the concentrations as a function of time, either in the discrete limit for stochastics or in the continuum limit for differential equations, or maybe you have some hybrid simulator that's something in between. And then you get the time courses of all the different species in the system as a function. The concentrations as a function of time. And in much the same way that we were able to make rules that described the molecules and their interactions, we can make rules that summarize the kinds of outputs we want to see. For example, you might be able to measure in an experimental context the amount of phosphorylation of a particular receptor site, or the amount of the extent to which a particular molecule is complex to another molecule. Those would represent different observables for the system. And there might be hundreds or thousands of species that correspond to those observables. And using the same language, the same BioNetGen language that we use to construct the rules, we can also use that to determine the outputs in a concise way. And we'll see examples of that. Okay, so I just wanted to go over briefly the main concepts that are here. So we have our set of molecules that we start with in our model description. And typically, so we need a starting point. We're going to generate all the possible species that can form all the different complexes and phosphorylation states of these molecules that are in my system. And we need a starting point for that. So typically, we take as the starting point the individual molecules in their ground state, their unmodified, unbound state. We call these the seed species. And then we start applying the rules to those seed species. So we have our, in this case of the IgE receptor system, 19 different reaction rules. We apply them to the species that are initially present. That generates new reactions in species. So for example, the ligand can bind to the receptor molecule. Then a second receptor molecule can bind to the ligand molecule. You have a dimer. The dimer binds Lin, and so on and so forth. And each of those different interactions that are possible represents one reaction, and new species are formed from that. And we keep doing this in an iterative fashion until we build up a large in this case, a network of 354 species and 3,680 reactions. And when we apply the rules to that set, no new reactions are generated. We're done. So we have convergence there. This is a closed system. Now, what you'll find many times when you specify a model, particularly if you go to the literature, write down lists of phosphorylation sites and binding sites, you may find that the model actually doesn't terminate, that this generation process keeps going, and when it gets to 150,000 species and some similar number of reactions, you run out of patience, or the computer dies, or something else happens.
So a really good example is polymerization
Yeah.
of actin from G-actin. How long of a filament can you generate? There's no limits, right?
Right, exactly. So right, a simple example is, if you can form a chain of molecules, then it doesn't terminate. And you have to give some guidance to the network generation process to tell it to stop after a certain size is reached, or a certain number of rounds, or so on and so forth. Typically, we don't use very often this network generation approach for systems that are non-convergent, which have infinities in them. We use another approach for simulation. So part of the reason I'm telling you about how this works is because I want to tell you how there are some pitfalls you need to avoid in simulation, and it helps to understand the basic idea of what we're doing here. So if we can form an infinite chain out of any of these components, this process is not going to terminate, and we're going to have to either tell the generator a way to terminate, so a number of times, size of complex to allow, or number of iterations to perform, or so forth, or we're going to have to take another way of doing this. And there is another way. So the other way is what we call network-free simulation. And the idea here is, instead of trying to generate all the possible species that you can get to, we just say, OK, stick the molecules in a box. This is kind of a virtual box in the sense that if we have a well-mixed system, the location doesn't mean anything. We just have a bag of molecules. It's a well-mixed bag. We could think of it being shaken up. And then we have our rules, which say what can interact with what. And the basic idea is that at any given time, the rules keep track of what reactions can take place in the system, and we sample those in a stochastic way. Basically, we assume that the reaction rate is proportional to the what is given by the individual propensities, those rate law functions that I talked about before. If we have mass action, then the rate of AB binding, for example, is the concentration of A reactants times the concentration of B reactants, some rate constant that we gave in advance. And we can then do a simulation where we just sample stochastic reaction events over all of the different rules for our system. And we never bother to generate all the possible species. We just generate what different configurations of this set of molecules can occur at a given time. And that's always finite, and we never deal with infinity here. And what's pretty remarkable about this situation is that this network-free simulation method has a scaling, a computational scaling. So this is the amount of time it takes to run a simulation of the system that is relatively independent of the size of the reaction network. So if we this is really the runtime per reaction, you could think of it. How long does it take to compute a set of a fixed number of reaction events? Well, as we make this network larger and larger, so this is log scale the number of possible reactions in the system, the network-free simulation method doesn't really care how many different possible reactions there are in a system that has combinatorial complexity of whatever magnitude. But all the other methods that we're showing here that are based on actually generating a network in advance, or basically unrolling that network as the simulation advances, they have a scaling which really hits a wall in the face of this combinatorial complexity. So the idea of the network-free is that we can build up a model of increasing complexity, include more and more components, refine our description of the molecules, for example, to include more mechanisms of regulation, or more phosphorylation sites, more regulatory molecules, and so forth. we can build that up without hitting a computational bottleneck as far as being able to simulate the system. So the basic then simulation methods that are available in BioNetGen are these. So we make our model of this system. This is the IgE receptor system with some extra stuff thrown in. We can then we pass this to BioNetGen. It can either generate a reaction network, which we can simulate using standard reaction-based methods, ODEs and Gillespie method, for example, or we can simply basically export this model to our network-free simulator and run a simulation. And we basically get the same outputs, the concentrations as a function of time here. This is a stochastic algorithm, so it's going to have some noise associated with the outputs, just like we would have if we ran a Gillespie simulation. But that's typically could be representative of the noise of a single cell, or it may be that we average that over a number of simulation runs to get a reasonable answer. So you have a question?
I mean, there must be interactions that we don't know about. Don't they have any effect on this? I mean, the result that we get.
Of course. I mean, well, so the idea is that here we might start with a very simple model of this system that has just some abstracted representation of the interactions, or we only say in our initial model consider these molecules, but then there are a number of other adapter molecules which bind and will change the behavior of this system. So the idea of the network free is to allow you to start with this model that might be here in this complexity space, add some components here, and add some more components here, and check how does the behavior of the model that has this complexity differ from the behavior of the model with this complexity. So I mean, the problem with the standard approach with reaction network modeling is not that it's bad to be abstract. It's not bad to be abstract. The problem is that it's bad to be abstract without knowing what you've left out, without having a way to test. It's like if you did, in MD world, this is almost like it's a problem that's hard to kind of imagine, because nobody would say to you as a molecular structure person, oh, well, you shouldn't consider molecules which have the full structure. You should only consider very highly abstracted models of 3 variables or less to describe your protein behavior, right? No one would tell you to do that. But for a biochemical network, well, we know the standard argument would be, well, we know so little about these systems that we could only possibly describe them by 3 ODEs or less. But we actually know a lot about the biochemistry of these systems. And so it really makes no sense not to have a reasonable null model that might lie out somewhere in this space and say, okay, if we know, based on the interactions we have for this system, based on what we know about the biochemistry, what does the model predict? And compare that to a much simplified version of that. Yeah.
The final goal of a signaling molecule is to elicit a physiological response. The cell is to elicit a physiological response, which, as you have indicated, goes to the binding of a receptor like in the growth factor and alters the receptor to phosphorylation and causing a cascade of biochemical reactions. And this cascade will probably influence the DNA and make it elicit a physiological response. So for growth factor, taking growth factor as an example, there is a termination factor involved. Once the physiological response has been elicited, there is a termination. And so it varies from reaction to reaction. As Tom just mentioned, there are some reactions, there is no termination, like reaction goes on. So in your model, what tells the model or your information that you're feeding that it should terminate or not terminate? How does it accommodate the information regarding termination?
Okay. I don't know where to start with it. I mean, my model is not about signaling or intention or physiological response. I'm just modeling chemistry. I just have I have a bag of molecules that interact with each other, and I claim that this represents some biochemical interactions that I know something about. But I haven't, for example, in this model, I agree, many things happen here. Many changes in protein expression happen here. mRNAs, microRNAs, soluble molecule, all kinds of things go on in the cell, only a small portion of which I'm representing here. And so far in our model, we only considered explicitly a few proteins. We know something about how they bind to each other in the phosphorylation events that can take place. We don't know what, quote unquote, terminates their interactions. Maybe ultimately the regulation of the expression of these proteins is regulated, and we can include all of that in a physical way.
Your model is excellent. I'm not criticizing
No.
the model. So what it is a promise to give you a lot of reactions. For example, Eric Kandel said, signal transduction is the key to understanding how diseases occur and how a certain cell makes order. So this model is able to answer that a certain growth factor, the absence or the presence of it, can lead to a certain disease, like cancer, for example, and it would be very interesting.
Sure. I mean, go ahead. Maybe you have a better take on it.
The short answer to your question is if there's something that terminates the signal, and you know what that is, then you need to include it in your model.
Yeah, that's what I'm saying.
Yeah. And that's the simple answer. And if you don't put it in your model, then the model won't terminate. It won't have that.
Let me show you something. So here's the logical consequence.
Yeah. Yeah.
Here's the logical consequence of the statement that you quoted. If we want to understand, so if we want to know what happens in a cellular response, we need to look at signal transduction. Well, this is a model that's involving. It's a model of T cell receptor signaling, and it's a little. I know it's very hard to read the individual components, but this is basically. Wait a minute, where did the receptor go? I have to take a step back here. This is the receptor, and this is the initiating kinase. This is the first layer of kinases that respond. What's represented here are all of the proteins that are known to become phosphorylated in the first 60 seconds after you stimulate the T cell receptor. We could make very similar model diagrams of the responses that are downstream of any receptor probably almost any receptor you pick in any system, any cellular system. Certainly that would be true for AMPA receptor and NMDA receptor. People have made similar kinds of studies using phosphoproteomics of the phosphorylation events. And so there's just this unbelievable level of complexity, which really, as you allude to, also only scratches the surface. This is only what happens in 60 seconds, long before you have any transcription, translation, and so forth. And so it's just, it's unbelievably complicated. And we don't really know at this point how much of this do we really need to understand a given response? How much of it contributes to the response, the decision of a T cell, for example, to become energetic or to become activated, just to throw out example of a physiological behavior. It's pretty remarkable that a system that's trying to make a simple binary decision, for example, uses all of this complexity. But this is a map of what we actually so far know. So the idea of the rule-based approach really is to just give you a language in which we can if we given a wiring diagram of this sort, we can make a model of it, and we can make a simulatable model of it, and we can ask it questions like, given an input of this magnitude, how much response do we expect at some time later on? But it has very severe limits on it because we precisely don't know what are these, how does the cell terminate that response? What are the mechanisms, that...
The cutting edge of research.
Yeah. Still, even though sometimes it comes and goes in terms of trend, the what's considered cutting edge and important. Now everyone, now the focus is on sequencing everything because we have the technology to do that very effectively. But there's a lot of biochemistry that's happening that's completely, it involves no changes to sequence whatsoever. So we have, we have a long way to go to understand all of this.
There's a mathematician who studies the mathematics of networks, not just reaction networks, but just interaction networks of any kind, graphical networks in general. His name is John Guggenheimer at Cornell University. And a number of years ago, he wrote a paper with a mathematical proof in it that proves that you cannot consider a subgraph of a network correctly in isolation from the whole network. This is a horrible, horrible thing. Right? But there are certain approximations that you can make under certain circumstances that if the assumptions are okay, then you can bound the error that you're making by considering the part in isolation from the whole. And some of the rules about that have to do with separation of time scales. So some things are happening very, very fast, and some things are happening very, very slowly compared to the rate of what's happening within your subgraph. Then you can put certain assumptions and boundary conditions. You can simulate the subgraph in the presence of certain boundary conditions, right? And that's basically, what you have to do, that we all need to do, right? Because we're all considering subnetworks of the whole network. What this really means is you can't even consider a single cell in isolation from the body. You can't consider a single body in isolation from the population of other individuals in the population. You can't consider the population in isolation of other superpopulations, right? It gets to be silly.
So,
you can't consider the Earth in isolation from the universe, right?
So given that, we'll just take our reductionist approaches as a leap of faith, and we're not doing science anymore. This is just an exercise in faith-based modeling. Okay. So let's be practical for a moment. I'm going to tell you a little bit about how we actually construct a rule-based model of a system, and the idea is that all of this is an abstraction, a small piece of a network. We can do experiments and find that these different components have some effect on some outcome that we're interested in, and we want to kind of understand how that might come to be. And that's where we start to use these kinds of biochemical modeling approaches. So RuleBender is a tool that we developed that lets you, it basically provides an environment for developing BioNetGen models in very much the same way that CellBlender, which is very hard to say, it's very hard to keep CellBlender and RuleBender straight. If you say that a bunch of times, you'll have CellBender and RuleBlender before too long. But that's fine, because actually the ideas are very similar. We want to have a model description and a visualization, and we'll see the results will show us the behaviors of the model, an environment for doing all of that that's self-contained and easy to use and easy to extend if you get into if you have certain capabilities that you want to add to that system. So RuleBender is a tool that we've developed for BioNetGen models, and I just wanted to show you a little bit about how we would construct a simple model using BioNetGen and how we would simulate that in RuleBender. So the basic components of a model are listed here, are a set of blocks. Very much like in MBL, you have blocks for the different aspects, the parameters, reactions, molecules, and so forth. We have the same thing in BioNetGen, and actually one of the goals of the BTRC, the modeling center that I talked about at the very beginning, is to develop a unified representation of these things that spans between MCell and BioNetGen. So these are the components. So in a BioNetGen file, each of these is separated in a separate block. We have parameters. I'm going to show you a description of a model that's compartmental, in which the different surfaces and volumes are explicitly specified, so that we can actually translate our BioNetGen model, in which these compartments are assumed to be well mixed. So there's no geometry. There's just a partitioning of space, if you will, directly into an explicit representation of space that we could simulate in MCell. So the model that I'll show you for receptor dimerization in BioNetGen, we also, Deepak will show you after the break in the lab how we translate that into MDL. So we'll go from a compartmental representation to an explicitly spatial one. And then the other standard components of the model are we have to define our molecules. We define these seed species, which is basically the starting point for our network generation, but it's the initial things that are present in our model, the initial molecules that are present in our model. They don't have to be only molecules. They could be complexes of any level of complexity we want. The things that we want to read out from our model that we call observables. Functions are something that we generally avoid for a model that we want to translate into MCell. These are going to be functions that allow us to do non-elementary reaction kinetics, which is common in well-mixed systems. So most of the nonlinear kinds of ODE models use some nonlinear rate laws to give rise to complex phenomenon. We're going to have to avoid those if we want to simulate it directly into MCell. It's already an abstraction. And then the rules, the reaction rules that describe the interactions between the different molecules. So all of this gets turned into a BNGL, BioNetGen language file, gets passed to BioNetGen, and then we can run simulations based on different approaches. And the simple model that I just wanted to present to you is one of dimerization, we could call it ligand-induced dimerization. We have our antibody chemically cross-linked, covalently cross-linked dimer here. So it has two binding sites that bind to the receptor. The Fc portion bind to the alpha subunit of this receptor, which is exposed on the cell membrane. There's some rate constants that describe that binding and unbinding reaction. And then once the ligand is bound to the cell surface, that's represented here, one site, it's effectively diffusing in a very much reduced volume on the plasma membrane of this cell, and it can then bind with the second rate constant to another receptor that's on the cell membrane, forming a dimer. And the extent of dimerization as a function of the ligand concentration is an interesting property to study. So we might assume, for example, that we have a ligand. It stimulates this receptor system. As we increase the concentration of that ligand, you'll eventually saturate the activation of the receptor. We might assume that. We'll see. We can test whether that's a correct assumption. And OK, so the physical, so the compartmental description. So this could be, we could describe this just as a set of two different reactions. It's actually 4 reactions, including forward and reverse, but 2 different rules for interaction. Independent of this spatial representation, we could just implicitly understand that the ligand is in one compartment, the receptor is in another, and then the ligand-bound receptor and the dimer are 2 additional species, and those are also in the plasma membrane compartment. But we could just dispense with the representation and just put the effects of the compartmentalization into the rate constants. But we won't do that here. We'll explicitly represent that the concentration of ligand is defined over this volume that's in the extracellular region of the cell. Then there's a plasma membrane, and I'm representing these as squares because one of the simple translated versions of the models that we have uses nested cubes. So basically, we have an outer container cube. The space between that outer container and the container representing the cell is the extracellular region. Then there's a surface mesh that represents the plasma membrane, and that's where the receptors are going to be located. And then there's a cytoplasm where we could put other stuff in here. We have a full representation of our IgE receptor model with its 354 equations, but we're not going to use that as our example here. Okay, so what we need to do is to represent this spatial, this compartmental topology in BioNetGen, and what's shown here is how this translation works. So we have now our BioNetGen block that begins with a begin block name and end block name, so begin compartments and end compartments. And inside that block, we have code that specifies each of the compartments here. So Typically, we go from outermost to innermost. And at each level, we have an object. Here, it's the outer container is called, we call it wall. The second column represents the dimensionality of the compartment. So in BioNetGen, you basically have two alternating 2D and 3D. So you have or 3D and 2D, but in this case, we have an outer container that's a surface, and so it's two-dimensional, and then it has some volume associated with it. It seems a little bit odd to talk about the volume of a surface, but what that really represents is that there's a surface area associated with this wall, and then we assume for the purposes of reaction diffusion that takes place on that surface that it has some effective thickness, which here we take to be 10 nanometers. And so that essentially tells us how to scale the concentrations of the components and their associated rate constants if they're given in standard units. I'll go into this in a little bit more detail later on. But so you can think about this, but the volume of any surface is simply its area times some effective thickness. And then, and that's it. So the last element which you see shown on these is the compartment that contains that element. So for the outer wall, there's no containing compartment, so we leave that blank. And now we go to the extracellular space, which is a 3D space and has a particular volume. And that volume in a standard, if this were a suspension of cells that we were representing, that extracellular volume would typically be taken as 1 over the cell density. So the amount of extracellular medium that a cell effectively samples or has associated to it is just 1 over its density, which is typically, say, in a suspension might be 10 to the 6 or 10 to the 7 cells per milliliter, and from that we can get an effective volume. Often when we do conversion, when we're taking a model that we're going to represent an M cell, we'll simulate only a small portion of an entire cell, because otherwise the simulations would be lengthy. But okay, so we have this extracellular volume that could be 1 over cell density, and its container is the wall. So its containing compartment is the wall. It's represented here. And then we go on to the plasma membrane. That's a surface. It again has a volume that's its area times thickness, and it's contained by the extracellular region. We could at any so if we think about this, there could be multiple cells. We could have PM, a plasma membrane here, but we could also have another plasma membrane that's contained within this. It's contained by the extracellular space, but it's distinct from the plasma membrane. So we can see that we can nest multiple surfaces inside one volume. We can't have multiple volumes inside one surface. So when we go from 3 to 2, we could have PM1 be inside of extracellular, and we could have a PM2 inside extracellular, and so on and so on and so on. But when we have a cytoplasm, we can't have multiple cytoplasms inside of one plasma membrane. Okay, it's just a fundamental topological relationship. We can. So what we're representing here is the topology of space. We're not representing what's the word I'm looking for here? We're not discretizing space. We're not subdividing either the membrane or the volume spaces themselves. So we're not basically forming a grid or something like that. For that, we need a more explicit spatial representation. Yeah.
Does the wall EC include the volume of the cytoplasm?
No, it does not.
It's just.
Yeah.
Okay.
That can be tricky. Right, it does not. And so that we have to take into explicit consideration when we actually construct the M cell geometry. Okay. Right. Questions about this? Is this relatively straightforward? We're representing a topology essentially as a tree structure here. And so all you need to represent a tree, for every element of that tree, that element needs to know its parent. And because it's a tree, you only ever have one parent. Okay.
I have a question. When you define,
Yes, absolutely.
when you define the volume of the PN, the plasma membrane, you define it's a surface with a thickness.
Yes.
The thickness is always 10 nanometers per volt.
It doesn't have to. I mean, I think that the way that we have the translator to MDL set up now, it assumes the thickness of 10 nanometers by default. That's something that we could configure, could change, and we could, the translator is sort of at the pre-alpha stage, is something that we're just basically playing with now. But in the general BioNetGen representation, this could be whatever thickness you wanted to represent. Now I have to think. I mean.
And you can also, because vol EC does not include vol of CP, you can construct trees that don't conserve volume. You have a surface area.
You could construct very non-physical things. Yes, you could.
So you're creating surface area to volume ratio.
Yes, you are.
And the minimum surface area to volume ratio is a sphere, right?
That is correct.
And
So in
you
principle,
could
we can check. We could do
construct
checks.
things that are nonsensical.
You have 3 minutes left.
Oh.
Yeah, it's a little large because
That's.
if you want, I'll have to switch the.
It's not a problem. It is a natural. We could naturally take a break, and do a demo. Do some of the demo stuff over lunch as we did yesterday. That might work well. I'm sure you guys have been sitting here for a long time this morning, listening to people talk, and could probably use a good break.
So I was
Yeah.
saying that
So let's.
you could construct things that don't add up to the
That is true.
that are smaller than the minimum surface area to volume ratio
We should detect.
applied by a sphere, and that would be
Now,
non-physical and bad.
I have to think. My brain can't do the calculations while I'm sitting here at the board. I have to think about the way that what the constraints are on these volumes, given the assumptions that we're making. Because here, any thickness could be assumed. So this says what the volume of those layers can be. It doesn't say what the area surface. I don't think it explicitly tells you what the surface to volume ratio is until you pick a thickness. If you make an assumption of D, then these are constrained. But if you don't, then I'm not sure. I have to think about that.
But in the limit as thickness goes to 0,
Yeah,
you
then.
have a sphere with a surface area that has a minimum, and you could violate that.
I think so. I think it's possible. So, I mean, and we always assumed that that the user would be, having some topology in mind, some geometry in mind, and give these based on that mental geometry. This is
Right. No, but there may be another way to construct this.
there could definitely.
Automatically correct.
That's in other words, if this, as you said, represents a total volume, but then you have fewer. I don't know how many. So here, for example, we have a topology and we have given 4 independent parameters, but I'm not sure that there are constraints, as you point out.
But they're not independent because
They're right.
they
Right.
don't sum. If you construct it differently, I think you can get it so that they have to sum.
Yeah.
And it would be the same number of constraints.
Let's work that out over coffee. Yeah, that's. Suffice it to say, you should try to make these as realistic as you can. If you make your computation based on a real geometry, this won't be wrong for sure. If you just pick arbitrary values there, you're subject to some mischief. Okay. Yeah. So this concludes the part before the tutorial, and I'll actually show you Rule Bender and how we construct this model explicitly in the software And we can go over some examples of specific rules in the syntax during the lunchtime session. And then we'll give a little preview of the translator, how we take a model like this model in particular, and translate that into MCell and do simulations.